So I'm trying to be provocative here with the question, what's wrong with neuroimaging research and how can we make it right? So people like to make fun of phrenology, right? Many of you are familiar with phrenology. This is this thing that people did in the 18th century. It actually was, the people who sort of first came up with the idea behind phrenology, like Franz Gall, he was actually a serious neuroanatomist, probably one of the best of his day. And so they had this idea that, different parts of the brain do different things. That was actually a pretty revolutionary idea that we still, many of us still walk around with. But they made this mistake by thinking that the degree to which a particular part of the brain is kind of good or bad at something would cause it to protrude enough that it would, kind of cause a little bump on the skull and that you could feel these. And so it turned into basically a pseudoscience. People would, go around sort of like fortune tellers reading people's heads. And so you might think, we have these amazing like multi-million-dollar machines. We have to be doing better than phrenologists, right? And you would certainly hope that's the case, but there's a lot of reason to be concerned about many of the practices that that many of us in science have used for many years. And this has really come to the fore in the last decade with what at least some of us refer to as the replication crisis. Others really recoil against that term. But sort of starting really in psychology, but then growing and sort of becoming clear in lots of different areas of science, it's become evident that many scientists were sort of really generating BS rather than knowledge, and that particular practices within particular parts of science were sort of more or less sort of amenable to really, generating ideas that weren't really reproducible or replicable. One kind of, I don't know how many of you are familiar with this, but this was a study that came out, I guess, in 2015. A number of psychologists tried to replicate 100 published findings in psychology journals and found that they could really only replicate, in terms of like finding another statistically significant finding, they can only replicate about a third of those. And that really lit a fire under people thinking, wow, look, we're doing things wrong. What are we doing wrong? Well, I'm going to talk through a number of the things that I think that we've been doing wrong. Now, we'd love to do something like this in neuroimaging, but going out and trying to replicate, doing well-powered replications of 100 imaging studies would be insanely expensive and labor-intensive. So that's just not going to happen, right? So the question is, how can we know how reliable neuroimaging findings are? Well, one way is just look at published findings and see kind of how well they fit with one another. So this is a meta-analysis published in 2017, actually sort of a re-meta-analysis of a number of previous meta-analyses of imaging studies of major depression. So these are all previous meta-analyses that had claimed that, well, look, you find replicable relationships between functional activity and diagnosis of depression, across subsets of people. That was 99 published studies that went into those meta-analyses. It turns out that when you do the meta-analysis correctly, there's basically no convergence across studies. There's no consistent difference in brain activity between depressed and non-depressed individuals, even though 99 papers had been published claiming to have found statistically significant differences between depressed and non-depressed individuals. That really makes you worried about how good the science is if you can get 100 papers published and not be able to find anything consistent between them. Another example from Fowler-Bocco and colleagues, this was the winner of one of the OHBM replication awards, where they tried to replicate 5 studies with 17 different correlations between brain and behavior, and basically found that they were really not able to replicate any of them very strongly. And there's a number of problems with this study, actually. It's not really a very well-powered replication study, but nonetheless, it suggests that we may have a problem with the ability to replicate published imaging findings. So it's a bit of a mess. How do we fix it? So I've given you the kind of, the storm and drang. Now I want to turn to kind of what can we do about it. And so we've I mean, this is something that's really consumed me for the last 10 years. I haven't really done that much science in the last 10 years. I've done a lot of meta science, and that's because I didn't want to keep doing science that I thought was just going to generate, false results. So we got lucky and got some money from the Arnold Foundation a few years ago to start what we call the Center for Reproducible Neuroscience. And it was really Chris Gorgolewski, who will be here, I guess, tomorrow, Wednesday. He and I started this center, and then he just recently moved to Google. And so the idea is, figuring out how can we actually fix the problem so that we can do science that we feel like we can actually have faith in. And one of the ways that I think about this is in terms of design. Science is a human creation just like anything else, like a toaster or this thing. Does anybody know what this is? If you've seen my talk before, you can't answer. Anybody have a guess what this is? Somebody must have a guess. It's an industrial something. Paper cutter. Okay. It has 2 green buttons on it. Why do you think the industrial paper cutter has 2 green buttons? Right. Exactly. So people won't do the stupid thing that people are really likely to do, which is to hold the paper in with one hand and cut their fingers off, right? So it prevents you. So the designers of this machine have anticipated the stupid things that people are going to do and built the machine to prevent people from doing those stupid things. And that's what design is all about, right? Figuring out how do you afford the behaviors you want to afford and disafford the behaviors that you don't want people to do. And people are really good at fooling themselves, right? This was a thing from Nature a few years ago. Humans are remarkably good at self-deception, and this ties in with we're just as good at self-deception in science as we are in anything else. And so one of the ways that I think about what we'd like to do in science is in terms of this idea of what's called choice architecture. This is an idea from behavioral economics, which basically is that any situation that people are put into, certain behaviors are afforded, certain behaviors are disafforded, right? When you walk into the store, the things that are right in front of the cash register are put there because you're more likely to buy them, as opposed to if you have to go dig them out of a tiny little place back in the back corner of the store. People like Thaler and Sunstein talk about this in terms of the idea of nudges, right? We can basically change the environment without removing people's freedom. You're still free to do whatever you want, but we can try to push you in the direction that we want to push you by things like incentives, defaults, feedback, predicting what errors people are going to make, and so on. John Ioannidis wrote this paper a few years ago that I think laid out a lot of the things that are wrong with science, and he was really sort of this paper predated, a lot of the kind of replication crisis stuff, but really sort of laid out the ideas that I think give us a handle on what's going on. So he talked about the various factors that drive a particular field of science to be more likely to generate false results. One of them is the smaller the studies, the more likely they are to the smaller the studies, the less likely they are to be true. Basically, the more things you're doing, the greater the number and lesser the selection of tested relationships, the less likely they are to be true. The greater the flexibility in the design's definitions, outcomes, and analytical modes, the less likely they are to be true. So this, in some sense, you can look at this and see these are all issues that, I think we have to grapple with in neuroimaging. So I'm going to talk about a few of these. First, I'm going to talk about sample sizes. So this paper came out a few years ago from Kate Button and colleagues, which analyzed studies both from imaging, actually structural imaging studies, and also rodent lesion studies, and basically try to quantify the power of these studies to find basically the effect size that you would have expected based on meta-analyses in that area. And basically they showed that the large majority of studies are badly underpowered. So a lot of the studies have basically less than 10% power. That means, right, even if there's a true effect, only 1 out of 10 studies is going to find that effect. So those studies are, in clinical trials, they have a thing they call futility analysis, right? To ask basically, is this study futile? At this point, should we keep doing it, or is there no way we're going to find something? These studies are futile before they were ever done. So you might say, well, that's fine. If you do a futile study and you don't find anything, then you don't find anything. And 1 in 10 times you're going to find something. But as long as you control for your level of false positives, then you're still only going to have 5% false positives. And that's true. but that's not necessarily the number that we really care about, right? So what Ioannidis showed in his original paper, and then they sort of repackaged it in the Button paper, is that power relates to something that actually we probably even care about more, which is what you call the positive predictive value. This is, if you have a positive finding, you find a significant result, how likely is it that that significant result is actually true? So here's kind of the intuition. Let's say you're doing a study where you have a detector that's meant to detect something, but unbeknownst to you, the detector is broken. So it's just putting out white noise, right? So you're going to find, let's say you have a false positive rate of 5%. You do 100 studies, you're going to find 5 positive results, right? All of those positive results are going to be false, right? So your positive predicted value is 0, right? There's no true results in there. And as, let's say that you were able to kind of gradually fix the machine, and let's say that there is a true result out there, the machine comes to be able to find it. Now, the proportion of those 5% are always going to be there, but now you're going to start having more true positives. So the more power you have, the more likely it is that a positive is actually a true positive. The other thing that they show, and this Tal wrote a nice paper about this a long time ago, is that when you have low power, anything you find is destined to be a really substantial overestimate of the actual size of the effect. This is called the winner's curse. So power, basically low power leads to unreliable science. And this is, you don't have to look very far to see this. You just have to look to the field of genetics. So back around 2000, people started doing a bunch of these studies where they would look at a single genetic polymorphism. Like, people differ in this BDNF Val66 MET place in the genome. And we think it relates to the function of the BDNF gene. And so they would say, let's take 50 people with one version of the polymorphism and 50 people with another version of the polymorphism and compare their brain volume. In this case, this was a hippocampal volume. And so what we see here is the x-axis is year of publications. These started being published around 2003. The y-axis is how big was the effect size that they reported in the paper. And the circle size is the sample size. So what do you see here, right? The small samples are the ones that are giving you the really big effects, right? These guys. When you start having bigger samples, suddenly the effect, and the early studies are the ones that mostly give you large effects, and all positive effects, right? It's not until later when, you start seeing negative effects, and especially as the sample sizes grow. So there's a couple things going on here, right? When you have a small sample, you're going to get basically wildly fluctuating results. Sometimes you're going to find a really large positive result. Other times you're going to find a really large negative result. If you throw away the negative results, as people in this case did, and only look at the positive ones and only publish those, we call that publication bias, then you're going to have this literature that's full of basically, highly inflated, potentially false findings, right? And here you see that the effect size with the big studies has now basically come down to 0. And in fact, now that we have these large, people in the genetics field did this for probably about a decade, and then geneticists said, hey, wait, none of these small studies of individual genetic variants are replicating. We need to start looking at the entire genome and controlling our statistics for the entire genome. So that means, instead of p less than 0.05, it's like p less than 10 to the minus 8th, right? So much more stringent correction. That means in order to be able to have enough power, you need tens of thousands of people in your study. This was the first of these GWAS studies on hippocampal volume by Stein and colleagues. And they basically said, look, there's a bunch of genes that people have claimed in the literature to have found associations with hippocampal volume for. When in a well-powered study, I think this was 2 groups, a total about 20,000 individuals. None of those are reliable. They find other genes that nobody had thought about that actually do have a reliable relationship with hippocampal volume. These ones that were sort of like one of my geneticist colleagues uses the analogy like the drunks looking for their keys under the street lamp, right? These are the genes we knew about, COMT, BDNF, whatever. So let's go look at them, of course. So how well-powered are fMRI studies? It may well be that that this doesn't matter. We did this analysis a couple years ago and looked at least up through 2015 that the median study in 2015 was powered to find an activation, like a single 200 voxel activation with an effect size of about 0.75. That means about 0.75 standard deviations between the baseline and the activation task. Now, if that's a reasonable effect size, if we can expect fMRI to give us effects of 0.75 regularly, then that's fine. That means we're powered to find the kind of effects that we can expect, right? Power is always in the context of a particular effect. If the effect is huge, right? Let's say that I want to do a study to ask whether 3-year-olds are shorter than adults. Right? I don't need a very big sample size to know the right answer for that. Right? Huge effect size you can find with a small sample. A subtle effect size you need, like those genetic effects, you need much bigger samples for. So the question is, what's a plausible effect size for fMRI? The problem is you can't just go to papers and look at the effect sizes that they reported, right? Because of the winner's curse, right? The effect sizes they reported are almost always going to be inflated, sometimes a little bit, sometimes a lot. So you have to play tricks to go in and find what are the effect sizes. And we used, Ariel mentioned the Human Connectome Project data. They've actually been a real goldmine for these types of questions. We used those. They had actually written down in the paper before the study was ever done, what are the regions of interest where we expect to find activation for each of these different tasks. So we took that, and then we took a meta-analysis from this tool called NeuroSynth that Tal built, and put those together to basically say, this is our region of interest. This is an area where we're pretty sure that there should be activity here for this task, like in the motor cortex for a motor task. Put those together, and that gives us a relatively unbiased effect size estimate, because these regions weren't found based on the data we're looking at. They were found completely independently. And it turns out that when you do that, the effect sizes are actually much less whopping than you would expect. So this is on the left is sort of how much the signal goes up within an individual. So we often quantify what we call percent signal change. It's like, if I have you resting and then I give you a motor task and then I have you rest again. And the activity in the motor cortex might go up 3 or 4%. And that's about, in fMRI, you never really see it go up higher than about 5%. And that would be in like visual or motor cortex. But that's not the effect size that we really care about, because that's within a person. What we care about, because we're doing statistics across people, we care about what's the size of the effect with respect to the variability across people. And that's what Cohen's d, the measure that I mentioned earlier, tells us. And it turns out that, so this is we commonly say like a Cohen's d of 0.8 is a large effect, 0.5 is a medium effect. And this is showing how big are these effects. So what this is saying is for things like the motor cortex in a motor task, which is about the most whopping activation you will ever expect to find with fMRI, it barely gets up around a large effect, right? So the effect sizes are about 0.7. So basically, half of the studies published in 2015 weren't even well enough powered to find activation in the motor cortex reliably. So let's go back and look at that depression meta-analysis. So what I'm doing here is, as a colorblind person, I should know better than using something that you have to see color for to pull apart. This is coloring what I'm calling grossly insufficient N, which is, this is this Simmons et al. Paper. The title is False Positive Psychology. It's actually a really great paper. They coined the term p-hacking. And they have a set of rules about like how to do better science. And one of them, this is particularly in the context of psychology, one of them is basically, it almost never makes sense to do a study with less than 20 observations per cell. That's not saying that 20 is enough. In fact, 20 is probably never enough unless you're looking at huge effects. But it's almost always going to be too few. And so I call that grossly insufficient. Right? This is coloring. So the x-axis is year of publication. The y-axis is sample size. This is basically saying the majority of the studies in that meta-analysis had grossly insufficient N. There's a couple of ways that I like to think about that. One is, so those were 634 sick people came in off the street to be in a study that was almost certainly futile. That was certainly, in fact, likely to generate insufficient power. Another way to think about it is, that's the equivalent, the amount of energy spent doing those MRI scans is roughly equivalent to what it would take for 44 cars to be on the road for a year, right? So that's wasted effort of sick people and lots of CO2 in the environment. Yeah, I know. There's a lot of assumptions. Some guy on Quora came up with a thing to do it. Yeah. So it's somewhere between 22 and 66 cars. Let's say that, right? It's a good bit of CO2. So, now those kinds of old school activation studies aren't cool anymore, right? You have to do something with machine learning. And you have to say, I'm not just showing that there's activation differences between depressed and non-depressed individuals. I'm going to classify, I'm going to predict depression, right? I'm going to have an fMRI biomarker for depression. Is Gael coming? Oh, no, okay. So if Gael came, he would give you a whole talk on this, basically showing that, this is some cool work he did, showing that, just as we saw in that genetics result right here, the larger the sample, the worse the classification accuracy gets for lots of different diagnoses. And again, it's not really surprising, right? Especially if you think about when people are putting together these kind of really complex workflows, they're trying lots of stuff. And in fact, I'll come back to this in a moment. And if you try lots of stuff, you're probably going to take the best one, right? Unless you've thought through what you're actually doing, you're going to try 10 things and take the best one, and that becomes the thing that goes in the paper, right? I'm going to try to convince you later that's a really bad thing to do. But that's how I think a lot of people have done science for a long time. So many of you you might have especially those of you who are, so maybe I can do a poll. How many of you are people who actually plan to do your own imaging data collection at some point in your career or have done it? Okay, so good. So I'm talking to nearly everybody in the room. And a question that often comes up here, right, is I'm trying to finish my PhD, right, and I'm in a place where I don't have money to collect hundreds of subjects. I can only collect 24 subjects. I can only collect 18 subjects, right? What am I supposed to do, right? And it's a really hard question, and I don't know that there's not a great answer for it, right? Because I don't think the answer is go do underpowered science, right? So I wrote this paper. Published in Neuron earlier this year, like a little opinion piece that tried to address some of these issues about what are the costs of reproducibility. So I go around giving talks telling people you need to do science that's more reproducible, but the upshot is if you do all the things that I'm telling you should do, it's going to make your life a lot harder. It's going to make it easier in some ways, like if you start using Git, it makes your life much easier, right? But if you start trying to do studies that are well powered, you're going to do less science. You're going to be more likely to find false null results. And so it's going to make your life harder. So I tried to talk in the paper about some of the ways to think about dealing with this. And I love this quote from Nancy Kanwisher. If you can't answer the question you love the question you can answer. So collaborate with other people. Find other people who have data that's interesting where they might not be doing the analysis you want to do and ask them if you can do that. Use shared data. The amount of shared data, as you heard, is becoming amazing, openly available data. Or focus on things like theory and computational modeling. I think we actually have a dearth of focus on theory in neuroscience in general, and to the degree that people start thinking more about that, I think that could be really useful. And just to note that Google and Facebook are hiring computational neuroscientists out the wazoo. So if this academia thing doesn't work out, you'll set yourself up well. So just one data sharing success story. So Max Schein was a postdoc in my lab from Australia for a couple of years, collected no data. So he's the ultimate data parasite, right? He's just sort of living off of other people's data. Published papers in lots of fancy journals, all based on shared data, right? All based on, not all openly available. In one case, somebody had published, actually 2 groups had published studies that used data sets that he thought would be relevant to ask a question. He contacted them and say, hey, would you like to collaborate? I'd like to analyze your data. That's the network neuroscience paper. Basically, we reanalyzed 2 other people's data sets. So there's lots of, sometimes it's openly available data. Sometimes it's contacting people and saying, hey, I'd like to reanalyze your data. Often that's going to require, some kind of collaboration. Fine. Okay, so let's turn back to the some of the other issues that Ioannidis raised, which I think are in some ways much more kind of within our ability to deal with. And this is things like the more questions we're testing and the less we're kind of selecting them, the more flexibility we have in our analysis, the less likely things are to be true. And this became really clear in a paper that came out, I think back in 2012, right, by Josh Karp, where he took a data set and ran it through almost 7,000 different analysis pipelines. And this on the left, you see the mean activation, which fortunately this was a data set we had published, and the mean activation didn't really controvert what we had put in the paper, so I was like, but this is a little scary, right? This is the range of Z statistics between like the highest and the lowest across those 7,000 pipelines. So this is basically saying there's 8 standard deviations of difference between results from one team or from one analysis and from another, right? That's a huge difference in results, which should really make you worried about what's going on between those different analysis streams. I'm going to focus for a moment on one particular thing, which is the degree to which you control for all of the different tests that you're doing. So generally, in an fMRI study, we're doing, let's say, 200,000 statistical tests. And if you don't control for those, you are almost certainly going to have inflated false positive rates. This is, again, with the colors, come on. It's showing in red studies that reported uncorrected statistical results. And you would hope that those would have gone away over time. Unfortunately, they haven't, at least not in this particular domain. And I just want to highlight that you can get, if you don't do the right corrections, you can get really beautiful data out of complete random noise. I generated a synthetic data set of 28 random subjects, fMRI data, 28 random subject behavioral data, did a whole brain analysis using what 10, 15 years ago, people would have thought this was a very reasonable thing to do. Just do p less than 0.001 uncorrected and only take things that have, say, 10 voxels in the cluster. I found this beautiful cluster with a correlation of random fake fMRI data and fake behavioral data of 0.87, right? And I didn't have to work very hard to do that. It was like, seriously, the first time I pressed the button and said, go, it found this. So the field actually has gotten quite a bit better. We did for the 2017 review, we looked at papers and said, what kind of correction did they do? And we found that of all the people who did whole brain analysis, only 3 of them were in that particular sample of papers. It was just like the most recent 100 papers or the most recent 100 data sets in the. This was activation studies reported in PubMed. So like the most 100 recent papers. But something weird was going on. And we had actually, Jeanette Mumford, who you'll meet later this week, she and I had been noticing this for a few years, hadn't really quantified it. So a lot of people would analyze their data with SPM or FSL, and then do the multiple comparisons correction using AFNI, using this tool called AlphaSim or later 3D ClustSim. And that takes extra work, right? It's not, you can't just say, go apply this, because you have to rename the files, or I think at some point you had to actually convert them to a different format. That's a problem, because basically it turned out when Eklund et al. Analyzed this that AFNI was doing some things wrong. It had a minor bug, but then it also had some sort of funkiness in how it estimated smoothing. And it turned out that it had a pretty substantially inflated error rate. So it's almost as if people had figured out we're going to get better results, meaning more activation when we use AlphaSim compared to when we use SPM or FSL's tools, which also had some issues, but not as big of an issue as AFNI. And so it shows that the people in the field seem to have been taking advantage of this flexibility that we have with all these different tools to actually do something that almost certainly inflated false positive rates. This is a great paper. I tried a bunch of things from Brad Weibel's team, which basically showed that if you just sort of randomly kind of, try different things. They actually built a genetic algorithm to try to increase the, basically try to change the features of a classification analysis to get classification results above chance when the data going into it were basically just sort of random, right? There's nothing to classify, but it turns out that if you take the same data set and torture it and try a bunch of things, you can find, just by kind of capitalizing on noise, you can find things that look like signal in the data. It's also not just fMRI. I think fMRI has been, the people in the fMRI world have been more sensitive to this for longer, but Steve Luck and Nick Aspelin had a paper showing that similar things go on in the ERP world, and I'm guessing that any field where there's complex analysis going on, not just in neuroscience, I'm guessing that in biology, there's probably very similar things going on when you're doing kind of high throughput, data intensive work, that the analyses become so flexible and the data becomes so complex that you can almost always find something if you're looking hard enough and not taking care to control things. So one of the things that we've certainly come around to is the idea of pre-registration. This is something that started in psychology a few years ago. And the idea is, it's pretty simple, right? If you're going to do something, write down what you're going to do and put it somewhere immutable before you do it, right? What's your sample size going to be? How are you going to analyze the data? What are the variables you're going to care about? This is an idea that really started in clinical trials back in the 1990s. And has really come to the fore in psychology. People are starting to do it in imaging. I think it's still not particularly common, but I think it's really, it's challenging for imaging because things are so complex. I think it's kind of getting back to something that you said about this kind of being a continuum. There's often a tendency to think that if I can't do it perfectly, I shouldn't do it at all, right? So like pre-registration, fully pre-registering an imaging study is really hard because it's so complex. You never know all the stuff you're going to need to do. But if you just write down as much as you can, you're already well ahead of where you would have started, right? So I think in general, like we often talk about like the 80-20 rule, right? Or not letting the perfect be the enemy of the good. And I think that's a good lesson to carry away from this, is like do whatever you can, even if it's knowing that it's never going to be perfect. Just do whatever you can. Also, another kind of misconception that people in the past have had about pre-registration is that you can't do any exploratory analysis, right? That you can't, let's say that you see something that looks interesting, but you hadn't written it down up front. This doesn't mean you can't go follow that thing up. It just means that when you write the paper, you can't claim that that thing was something that you had initially meant to test. And people can often even convince themselves, oh, well, I really would have thought that was something that I should have looked at. Pre-registration kind of holds your feet to the fire and says, look, when you're writing the paper, you have to be very clear. These are the things that we hypothesize. These are the things that were exploratory. And then the reviewers can take that as they will. And we don't have a lot of experience with this, but haven't found it to be kind of terribly problematic in the review process. Another thing that we've done is kind of multi-stage pre-registrations, where we first write down what's the sample size going to be, what's the data collection plan going to be, and what is our plan going to be to determine the analyses. So for example, we collect a data set, we use part of it to figure out what our analysis strategy is, and then we go write the pre-registration for what we're going to do with the other part of the data. Things like that, or I think that we shouldn't view pre-registration as like a straitjacket that you have to fit into. This is pretty amazing. So this is an analysis from a paper a few years ago looking at what happened after pre-registration became required in clinical trials. So in 2000, the Heart, Lung, and Blood Institute at the NIH basically started requiring clinical trials of drugs and treatments to do a pre-registration state. What's your end point going to be? What's the thing you're going to measure that you're going to kind of determine success on or not? Before that, people could measure, they could think we're going to go in looking at, say, all-cause mortality, but if they don't find something there, but the number of heart attacks went down, or some other measure changed, even though that wasn't the thing they really thought should change. So this is basically lower is better here, because it means that the person in the treatment has a lower risk of whatever the outcome is than the person in the control group. And this is the year that pre-registration started. Before that, these are all the positive findings, the ones in plus. So basically, almost every study had a positive finding, right? At least numerically, and most of them had statistically significant findings. None of them showed significant harm. After 2000, now you have 2 with significant benefits, 1 with significant harm, and most of them are basically sitting here around 0. Right? So it really changed what the results look like. So another, question.
For the OSF pre-registration, is there a policy regarding not reporting after you pre-register the study? So you pre-register something, it doesn't work, so you just kind of drop it?
So the question is, what happens if you pre-register a study and it doesn't work? Which means, I assume that means you find nothing statistically significant. I mean, in the best of all worlds, you would hope that any study that for any study you do, the result is important enough that there should be a paper telling what the answer was, positive or negative. But we know that the incentives for publishing null results are weak. Journals don't want them. Blah blah blah. There's no policy, I think, that says you are supposed to, if you pre-register something, you're supposed to publish it. One way to think about it is, people often talk about the file drawer problem, right? The idea that people would do studies and then stick them in the file drawer. At least here, the file drawer is public, right? So I can go on and ask, how many studies of ego depletion have been done, and I can, that have been pre-registered, and I can see them even if they never get published. I could, contact the author or whatever. So it's not perfect for sure, but I think it certainly helps to open up the file drawer a little bit. Another thing that I'll talk about a bit more is the idea of sort of using well-engineered standard tools, right? So the example from our work is this thing called fMRI prep. So, many different groups have different ways of pre-processing their fMRI data, and usually it was determined by whoever the postdoc was who showed up in the lab who knew how to analyze data, and they had come from somewhere that used SPM, so they become an SPM lab, right? Or maybe the lab next door uses FSL, so they're going to become an FSL lab. And that's problematic, one, because it's not optimizing for the best tools, and it also probably means that none of those individual tool chains have really been well engineered for quality control, and we'll talk more about software engineering in a bit. And so fMRI prep attempts to basically take the best parts of each of those tools using, I don't know if you'll hear about Nightpipe. Nightpipe is kind of the glue that holds all this together. And we think it sort of helps solve some of these problems of analytic flexibility. Okay, so let's turn then to talking about software engineering. So how many of you have written computer code in the course of your research? Probably if you're coming to this thing, you almost certainly have. How many of you have been trained in software engineering? Okay, noticeably fewer. So another thing that we have to worry about, and we know that any software has errors, right? So analyses of the software that run space vehicles, that sent the shuttle to space, have shown that even that code, which has been incredibly vetted, still contains bugs, right? And this can impact science. So this guy Jeffrey Chang was a very well-known structural biologist at Scripps, I think, published fancy papers in fancy journals, right? 3 science papers, some papers in other journals, talking about the structure of this particular protein, the ABC transporter. All of which had to be retracted because they had gotten a piece of software from someone else, and it had one flipped sign in it that totally changed the structure of the protein. It changed the answers that they had gotten from all these papers, right? So they ended up having to retract 5 papers in total. So I wrote a blog post a little while ago talking about sort of my experience with this sort of thing, where we had been trying to do classification. We had people doing a bunch of different tasks. We want to say, can we classify what task you're doing when we've never seen your brain before, right? We've seen other people doing the same task but never seen your brain before. It's like it's a hard problem. It's like 21 out of 23 tasks each person is doing, and we're claiming we want to know how well can we classify which of those 23 tasks you're doing. So we do this cross-validation. It's a little bit of Python code, and I got 93% accuracy. I was like, wow, if it had been 100%, I would have known I did something wrong. But it was 93%. I'm like, wow, that's really good. Right? Something cool is going on here. So at this point, I had a postdoc in the lab, Sanmi Koyejo, who actually has training in computer science and software engineering, he looked at my code and he's like, you did something wrong. You forgot to properly put this little indexing thing in, so you were basically flipping the matrix the wrong way. And when you do that properly, just adding those 4 characters, it changes the accuracy down to 53%, which is still well above chance, right? And fortunately, the paper was actually being written when I was doing this, hadn't been submitted yet, so we found it, luckily. I'll come back to talk about code review later because I think that's really important. But just to highlight the fact that really tiny changes can have big effects. And I think a big problem that many people don't acknowledge is the fact that this interacts with incentives around publication, right? So I use this term bug hacking, right? Which is kind of like p-hacking. The idea is like you're doing a bunch of stuff. There's going to be bugs in your software. The ones that you're likely to find are the ones that are going to cause results to go in your direction, right? If you have a bug that causes things to go in the other direction that you don't like, you're going to be more likely to find that bug, right? And this sort of PhD comics thing kind of shows that. So I wrote a blog post a while ago talking about this idea of the principle of assumed error, which is that if you find a result that looks good, try to figure out what you did wrong. And this is not a trivial undertaking, right? I have a postdoc in the lab who he'd been working with another postdoc, and they had found something that they thought was really cool, a result in a data set they collected. But one of them just had a funny suspicion that something was wrong. And so he played around with the data for at least a month, trying different things, digging into the code, finally found an error in like how they had coded something on the behavioral data, like before it even went into the fMRI analysis, redid it, and basically the result fell apart, right? Because it was based on a confound. And so it's bad for his career. If he hadn't done that, he probably would have gotten a paper in a reasonably good journal. He would have a first author fMRI paper on his CV, and that would have been really good for his career in the short term. I think in the long term, we have to think about what one of my colleagues calls the career-wise type 1 error rate, right? You don't want to be somebody who's generating false results because people come to know who the generators of false results are. Okay, so one of the things that we've tried to implement more, and I've tried to implement it more in my personal coding, and it's hard, is software testing. It's hard because it slows you down. It's probably good because it slows you down, but it's hard because it slows you down, right? Because you have to do stuff that isn't going to it's not on the direct path to answering your question. It's on this side path of like making sure that things are working. And there's lots of different types of software testing. And if you go pick up a book on software testing, it's probably going to tell you how to do kinds of testing that aren't really very useful for us as scientists. So what we often do is, what we call smoke tests, which is basically just, does it run? Does it crash, right? And this is useful. I'll talk in a bit about sort of automated testing. It's useful to be able to just say, here, I wrote this code, I submit it to GitHub, and then I'll get an email in 20 minutes basically telling me, hey, everything's good, or hey, you broke something, it crashed. Another thing is sanity checks. It's just like making sure that things are in the reasonable range. So this is a paper that was published a while ago and then had to be corrected, not exactly retracted, but corrected, because it turned out they had made these claims about like interactions not being significant, and it turned out that the data set included 2 notable age outliers, ages 5 and 32757. Right? We're pretty sure there's not a 32,000-year-old human, and it turned out that the results changed when they fixed that. Right? So all they would have had to do is put this into their code if they were working in Python. They were probably working in some like point and click thing where it becomes much easier to make errors, or at least it becomes much harder to know whether you made an error. If they had just said, hey, let's make sure that the ages are in the range that they're supposed to be in, and then the software would have errored out, right? So it turns out that it's, you can do this stuff on your own code. And once you start using Git and GitHub, regularly, it turns out that there's a bunch of tools you can use to help basically kind of automate this sort of testing. There's these platforms, like we use CircleCI, Travis is another one, that for open source software are basically free. At least there's limits on like how many hours you get every month, but we, for most projects, don't go past that. So the idea is that every time you commit a new piece of code to your GitHub repository, these tests get run automatically, and you will get, a notification or an email, however you set it up, telling you basically, did you break something, or are things still working? That requires that you've written tests, to find the things that could potentially be wrong. It's far from foolproof. It's easy to write tests that aren't good at finding problems, but nonetheless, it's better than doing nothing. Another thing you can do that I basically just started doing this month, I've become like a new zealot about this, is analyzing your code. And I'll tell you a bit in a few minutes about how I got here. So there's a bunch of tools that can basically take a piece of Python code and look for various problems in it, right? It's tools like Pylint, Flake8, that look for a couple things. One is look for kind of obvious errors, like, are you calling some variable that hasn't been set, or some function that you haven't imported, or something like that. But it also does something where for Python, there's a really explicit guide to not just exactly what you have to do, but what you should do in terms of how you format your code, right? So things like there should be, you should have spaces around your equal sign, right? Or you should have a space after a comma in this particular case, not in this other particular case, right? And that might sound like a really kind of annoying thing to have to do. And if it's only you looking at your code, that might be true. I'm not even convinced that's true. But certainly if other people go to look at your code, then it becomes really important to have done that. So I had sent the code I'm going to talk about for this NARPS project, I had sent to somebody who we engaged to do code review. And the first thing he did is wrote back and said, you need to do linting on this code. You need to fix the formatting. So I took about a week, went back, did all this stuff, gave him the new version. He's like, wow, the code is so much easier to read. I can actually see what you're doing here. And so this is like, the kind of stuff that it finds. You indentation with tabs instead of spaces, blah, blah, blah. All sorts of stuff like that if you want to write good Python code, you should be paying attention to. And another thing this does is, like, if you have a really complex analysis that's going to take 5 minutes until it hits the point where you put in a variable that is misspelled, you can learn that in 5 seconds rather than in 5 minutes, right? By just doing this, every time before you run something, you just run Pylint or Flake8 or whatever, and it'll find those, at least the big stupid problems. Like, I'm always misspelling stuff. This is maybe a little controversial, but I think that it's important. If Gael was here, this is another thing that he would sort of harp on, is basically whenever, don't reinvent the wheel. Scientists and especially engineers have this tendency toward what people call the NIH syndrome, not invented here, right? They think that anything that anybody else did is not good enough for me. I have to write my own new DICOM to NIFTY converter, even though there are 12 of them out in the world that work perfectly well. And rather than going and fixing the things that are wrong with number 11, I'm going to go write my own from scratch, right? And I'm going to write it in some, in Haskell or some language that nobody else can ever use. And what does that do? That means that nobody is you're going to be the only person who's finding those bugs, right? And whereas if you're using something that hundreds of other people are using, you have hundreds of other pairs of eyes on that code finding problems with it. And if you find a problem, you can, if it's a good open source project, you can just try to help fix the problem. One, you can report it, and two, if your skills are good enough, you can actually try to fix it. Submit a fix to the repository. And then another thing you want to do is make sure if you're using, when you're buying in, we're trying to figure out which of these tool sets do I use, right? There's 8 different toolkits for doing machine learning analysis in Python. Which one do I use? Well, one good question to ask is, are they doing good software engineering, right? Are they doing code quality testing? Or do they have good test coverage? Is all the code actually being tested? Those sorts of things. So this is an example from scikit-learn. I'm going to talk about badges later, but this is all of scikit-learn's badges, telling you all the kind of good stuff that it's doing. So I'm going to talk through for a few minutes an example from something that's fresh in my mind because it's something we're working on right now. I don't actually have results to show you. I'm going to focus more on process than results here. So this is this thing called the Neuroimaging Analysis, Replication, and Prediction Study, NARPS. Was anybody in the room actually on one of the NARPS teams? Okay, excellent. So we collected a data set and gave it out. We gave it out to, I think, 84 teams. And it was a complex fMRI data set, like 100 subjects doing this task, pretty substantial data set, a lot of work to go analyze this. And we basically said, we want you to go test 9 hypotheses. And those are like, there will be activation for this contrast in this brain region. They were all hypotheses where they just give us a yes/no answer. We're still working on the analysis, but it's pretty clear that there's a lot of variability across the answers across teams. And so we get their answers for each one, yes or no. And then they gave us the maps that were generated by their analyses, both thresholded and unthresholded statistical maps. And so that's what we're analyzing now. And this was an incredibly complex analysis workflow, right? Because we're taking data from a bunch of teams, running through all these different analyses. And there's both kind of practical and theoretical reasons to want this to be fully reproducible. Like I always want to be able to just punch a button and run all of my analyses from scratch. In this case, because, well, a few teams realized that they had done something slightly wrong and they gave us a new set of images, right? Suddenly I have to rerun everything. If I can't just punch a button and rerun everything, then that's just a bunch of work and more chance for human error. So I'm going to talk through a number of the things that we've been trying to do in this project to enhance reproducibility. And it starts with the data, right? So the data we wrote a data paper. I don't know if many of you are familiar with the idea of a data paper, but it's basically a paper that describes a data set. So there's a journal from Nature called Scientific Data there's other journals as well, that will publish basically a description of a data set. And so we have a paper that describes the data set. And then the data are shared through this project that we have called Open Neuro. And one of the things we do on Open Neuro is before a data set can be shared, it basically receives a version. It's like a snapshot of the data set. So this would be version 1.0.4 that was used in the analyses for the paper. That's the data set that we shared with everybody. So now if somebody else wants to go back and do something with it, they know exactly which version of the data set was used in the analyses. They don't have to kind of figure out what changed when. It's done, and it actually has an identifier. I'll come back and talk about identifiers later. We also share the data you're going to hear more from Kirsty, is that tomorrow about bids? At some point you're going to hear about bids, Wednesday about bids. So bids provides us a way to organize the data such that people know what they're looking at when they see the data. So you'll learn more about it. I'm not going to really talk about it here. You're also going to learn at some point about Docker. Docker turns out to be an incredibly important tool for building reproducible analysis pipelines. It's not the only way you can do it, but for us, reproducible and portable, right? So I didn't just want to have something that I could rerun on my own machine here. I wanted to have something I can give out to the world and say, hey, if you punch the button, you will get exactly the same answers. And Docker lets you get as close as possible to doing that. It's not always perfect, but it's pretty good. And we take inspiration from this quote from Buckheit and Donahoe. An article about a computational result is advertising, not scholarship. The actual result is all the stuff it took to actually generate the finding, right? Including the code and including the software environment. We know that the same code run in different software environments, different operating systems, even different versions of an operating system can give you substantially different results. Containers provide a way to share an entire computing platform, right? Because they encapsulate both the software that you care about, FSL or FreeSurfer or whatever, but they also contain all the libraries that are kind of lurking there in the background. So if you've ever had this thing where like your computing center says they're doing a software upgrade, right? And they don't tell you everything they're changing, but if they're changing like, glibc or something in the background, sometimes those can actually make a difference in terms of the results. This makes sure that whenever it gets run, it's run with exactly the same versions, as long as you and I don't know if Anisha will talk about this, or Chris, I'm sure Chris will talk about it. As long as you've made sure that exactly the same version of those libraries is being installed, right? It takes a lot more work to do this, but it ensures that if somebody reruns the thing, they're going to get as close to exactly the same results as is numerically possible. You may end up talking about R here. I mean, R, I have a love-hate, heavy on the hate relationship with R. It's a horrible programming language, but it has some tools that you just can't get anywhere else. If you want to do like, mixed effects, linear models, it takes a lot more work to do those anywhere else. It's not like you can't do them, but the tools that are most commonly used are, just so much more available there. And it's really hard to generate a reproducible R analysis. I did, for this myconectome project I did a few years ago, I gave out a virtual machine, which is kind of like an earlier version of what Docker does, that tried to reproduce all the analyses. And about a year later, I got an email from somebody saying, oh, hey, I reran your analysis, but now you found 8 clusters, and now I'm finding 10 clusters. And it turned out that, there was an upgrade in one of the deep down hidden versions of an R package where they changed something, it's giving different answers. But now there's a tool in this is what we use, this thing called Checkpoint, which will at least basically let you say, give me the versions of all the packages that were up to date on date X, like, July 16th, 2019. And it'll go on, this is from our friends across the water at Microsoft. It'll go in and basically download all of the packages that were up to date on that date. Now, the cost of this is you have to sit there and wait for it to install these packages. So, about a third of the time for my analysis platform to run is basically installing R packages for like one small set of analyses. But it's worth it to us because we want to be able to reproduce things. Makefiles are kind of a boring thing, but if you don't know how to use makefiles, they're certainly worth learning how to use. So this is basically a file that lets you set up a bunch of kind of common, if you're working in Unix, set up a bunch of kind of common commands. And so now instead of like typing this like nasty Docker command, I can say make run all, right? And importantly, I've set up my code checking. So before I go run it, I say, hey, make check style. In fact, I could just have it automatically run make check style each time and just it would crash if there was anything wrong with the style. I find makefiles incredibly useful. Your mileage may vary. I mentioned automated testing. This is basically all it took. This is my config file for Ccalm. So this is all it took to basically make Ccalm do automated analysis on my code. It's basically has to install some stuff. It has to run. It has to do the tests on the stuff in this particular directory. It runs my style checking, and then it does this thing to tell me how much test coverage I have, which is basically how many lines of code are actually being tested by the tests. And so I just stick this in my main directory, and then I go tell Ccalm to watch that GitHub repository. And now, anytime it gets pushed, those tests get automatically run. And you'll have a dashboard like this, which will tell you, oh, hey, this one failed, this one succeeded, and so on. Really handy for kind of, just making sure that in the background things are just always getting tested, so you always know if you've, at least after a little bit, if you've broken anything. Another thing that I just recently discovered, which basically this is I'm sure there are other tools like this. The one I'm using is called Codicy.com, which basically is sort of like taking things like that style checking stuff and kind of wrapping them all together and doing it for, like, if you have R file, actually I don't think it works for R, but like markdown files or HTML files or whatever, it sort of looks and finds all the different issues that you might have with different parts of the code, and tells you, oh, hey, you have these 17 security issues, 5 co-style issues, here's an error proneness thing, tells you what those issues are, and then you can go fix them. I'm going to skip test coverage. So one of the things, the reason that I started doing all this, I already mentioned, is because the person I enlisted to do code review for the project, when he first looked at the code, he said, hey, you need to do something about the style. I went and did that. But then he went through after I'd done that and started looking at what I had done, not just to say, you know, are there stupid bugs? But does the logic make sense? Am I doing anything that like doesn't make sense here? Also looking for stupid mistakes, but largely basically saying, are there any, like the little mistakes are often easier to find, right? Because they're usually going to cause errors. It's the really big mistakes that are often harder to find. And so I just had him submit issues on GitHub, and these are just a number of, if you go to this, there's a link to that NARPS page. You can actually see what the code review looked like if you want to see all the things that he found. Some of which are what he called nitpicks, some of which were a little more substantial. And then another cool thing about using GitHub is you can create sort of what they call releases, which are basically like snapshots of your code. Just like we create snapshots of the data, you can create snapshots of the code. So now when you write the paper, you can say, analyses in this paper were done using release 1.3 of this code. And you get a digital object identifier, which is something that can point directly to that code where it's going to live. We put it on this place called Zenodo, which is a place where you can sort of, you know, host random files. So now you know not just exactly what data went into the analysis, what code went into the analysis. And one of the fun things you get out of this is badges, right? So all these projects will give you this little bit of stuff you can put into your site. So now anybody who comes to your site will know that that you know you've registered it with Zenodo, that your tests are passing, you have 69% coverage. And when I started out, I was getting a C from Codacy. It took me a few hours of work, and I got it up to an A, which was fun. So you can get, like, you know, an A badge for your code. Now, obviously, one of the things that I probably don't highlight enough is, like, reproducibility is great, right? But something can be fully reproducible and completely wrong, right? Reproducibility is not the same as validity. So in psychology, we often talk about this distinction between reliability and validity, which is sort of a similar distinction. The idea is it getting, so reproducibility lets us know, is it getting the same answer every time? Validity means, is it giving us the right answer, right? Is it numerically right? Is it conceptually right? And that's a much harder thing to test. So one of the things you can try to do is basically put in data where you know what the answer should be and make sure you get the answer back out. Another one, which is particularly useful for any kind of machine learning analysis, is put in random data from the very beginning and make sure that, you know, do exactly the same process you did with the real data, and make sure that the data come out not telling you have 83% classification accuracy. And I think this stuff is best done in advance and pre-registered. This is like, do what I say, not what I do, because we haven't actually done this yet. We haven't pre-registered it. For the NARP study, after I had written all of the code, I decided, hey, we really want to, there's a few weird things. I wanted to make sure that they weren't bugs. So I basically generated data that looked exactly in their structure like the original data, but I knew exactly what the structure was. I said, you know, I'm going to take a few of the teams and I'm going to flip the sign on the data. I'm going to take a few of the teams and fill them with random data, and then I'm going to change noise across teams. And basically, now I know what the results should look like coming out. And the question is, do I find that? And so one of the things that we could do is just basically, you know, put in the data, run all the analyses, and then we get, you know, like these maps in the end, and we want to make sure, hey, is the map of the result at the end correlated really well with the map that was used to generate the data in the beginning, right? And if they're all correlated like 0.95 or higher, then I'm pretty happy with that, because there's obviously some noise. Okay, so to conclude, I mean, I doubt I needed to convince anybody here that we have a reproducibility problem, and I don't think we're alone. I think we're just tackling it harder than a lot of other fields. But I think that the complexity of our workflows and the complexity of our data sets certainly makes it more challenging and more likely to have those problems. Obviously, statistical power is a key, but that's a key that's often not as readily at hand in terms of being able to solve it as better software practices that'll let us make reproducible tools, and I hope that this has given you a glimpse into how many of the things I talked about today are tools you're going to learn about this week. And if you incorporate them, it's going to take you a little bit longer to write your code if you're going to write tests, but ultimately you're going to be doing better science. Finally, just want to thank all the people in the lab, particularly Chris, who you'll see later this week. a ton of amazing collaborators, and plug for my book if you haven't seen it. So, thanks.